Hello everyone, welcome back to my channel. And today I am going to discuss a very important topic of Cambridge A-level biology, which is light microscope and electron microscope and major unit conversions. So, in this lesson we will cover the structure of light microscope and transmission electron microscope, and we will also compare the both and the unit conversion used in microscopic systems. So these concepts are extremely important because Cambridge examiners frequently ask comparison and calculation questions from this topic. So let's start with the compound electron microscope. So actually this is a compound electron microscope. So if you look at this diagram, you can see different labeled parts of the compound light microscope. So let's understand the function of each part. The first part is you can see at the top of light microscope, we have eyepiece lens. And this is also called ocular lens. So this is the lens that we look through to observe the specimen or an object. It usually provides 10x magnification. So the image that we see through the microscope is further magnified by this lens. Below the eyepiece lens, we have another lens which is called objective lens. So, objective lenses are the most important lenses because they produce the primary magnified image of the specimen. Usually, there are three types of objective lenses. The first is low power, which is 4x. The next one is medium power, and it is usually 10x. And the last one is 40x, and it is a high power objective lens. So, these lenses actually are attached to a rotating turret. You can observe here, this is a rotating turret. So, it is also called a nose piece. What its function? It actually holds the objective lens and rotates to change the magnification. Suppose we have to change from 4x to 10x. We rotate the turret help. It will hold the rotate the turret to change the magnification. So the next we have the stage. You can see that we have the stage and this is the flat platform where we place the microscope slide. You can see that this is a microscope slide. This is microscope slide. So it contains the specimen or an object. The specimen must be a thin and transparent so that the light can easily pass through it. So the next is condenser. You can observe it. This is a condenser. And what is function? It actually focuses light on the object or a specimen with iris diaphragm. And iris diaphragm is used to vary the intensity of light that reaching the object. Iris diaphragm actually controls the amount of light that reaching the specimen. If too much light enters the microscope, the image may become too bright. So the diaphragm has to adjust the light intensity. So the next one is coarse focus knob. And what is function? It is used to focus the low and medium power objectives. Actually, this knob moves the stage up and down quickly. We can move the stage up and down. And it is mainly used for initial focusing when using low power objectives. And then the next one is fine focus knob. Okay, here this is a fine focus knob. And what is function? Used to focus the high power objective. It is mainly used when observing specimen under high power magnification. So at the bottom of the microscope, we have a light source. So this is a built-in light source. So this provides visible light which passes through the specimen and enter the lenses. Okay, this is the this is all the labeled part of the compound light microscope, and we have discussed all the parts. So now I want to just give an idea of visible light. So actually light microscope uses visible light And visible light is actually the portion of the electromagnetic spectrum that can be detected by a human eye. We can only observe or detect visible light. So the wavelength of visible light is 400 nanometer or to 700 nanometer. So the light passes through the specimen and is focused by lenses to produce a magnified image. But there is a limitation in light microscope. And what is limitation? Visible light have a high wavelength, longer wavelength. This is a wavelength, and it has longer wavelength. When there is a high wavelength, then we have low reducing power. Sorry, resolution power. And this is the main limitation of compound light microscope. And now I just want to give you an idea of resolution power. What is resolution power? Resolution power is actually the ability of an optical instrument to distinguish 2 points. Means we have these 2 points. If we have high resolution power, then we can distinguish between two. But if we have low resolution power, we these 2 points observe at just like one. You can't observe these 2 points. So, and it is a limitation of light microscope. Okay, now moving towards the electron microscope. In today's video, actually electron microscopes are of two types. The first is scanning electron microscope and transmission electron microscope. But today I am going to discuss a transmission electron microscope. So now let's transmission electron microscope is also called TEM. Usually, unlike light microscope, TEM uses uh, uh, a beam of electrons instead of light. It uses a beam of electrons. Let's understand now. Let's understand the part of this microscope. The first, at the top of the electron microscope, we have an electron gun. So the electron gun is actually emits an accelerated electron beam. Electron gun actually produces a beam of high energy electrons, and these electrons have to travel downward to the to the microscopic column. It moves toward. It has to move downward. So the next one is next part is condenser. It is also called electromagnetic condenser lenses. These are not glass lenses like those in light microscope. Instead, they are electromagnetic lenses. These are electromagnetic lenses which use magnetic fields to focus the electron beam onto the specimen. So the next one is specimen position. The specimen is placed on the path of the electron beam. And the electron beam has to pass through this column, and we have to place the specimen at this position from where electron beam has to pass. For TEM, the specimen must be extremely thin so that electrons can pass through it. So, but the main point is the specimen must be dead in the electron microscope, because electron microscope operate in a vacuum. You can say that where we have a part airlock or the specimen is introduced without the loss of vacuum. So this is an important point. The next one is objective lens. So after electron pass through the specimen, they enter the objective lens. These are objective lens, and it focus the image. So this lens focus the electron to form an image of the specimen. This is the main objective of objective lens. So another lens which is used in lamp is a projector lens. So it in the what its function? It actually further magnifies the image. It actually further magnifies the image. So here is a viewing port with a binocular viewer. The next below the uniform, we have a fluorescent screen. And the image is projected onto a fluorescent screen or camera. Here, the electrons are converted into a visible image that scientists can observe. So, at the last, we have a camera chamber, and it allows a black and white photographic image to be made. In transmission electron microscope, we get black and white image. We don't get colored image. So this is all about transmission electron microscope. So now let's quickly move towards the differences between both. I'm going to summarize the key differences. So the first one is we use visible light. As I already told you that a human eye only detect the visible light. So light microscope observe visible light to observe specimen. So in electron microscope we use beam of electrons. The magnification power of light microscope is about 1500x, while electron microscope is 5 lakh or more. Means electron microscope have a high magnification power, while light microscope has low magnification power. And the resolution is about 200 nanometers. While in electron microscope, the resolution power is 0.1 nanometers. And this is a huge difference because electron microscope has um, low wavelength, means shorter wavelength. Shorter wavelength. So it has high resolution power. Means if we have these 2 points, we can distinguish by using electron microscope, but we can't distinguish it with light microscope. We can't under light microscope we cannot distinguish these 2 points. So that is the difference. In light microscope, the preparation of specimen is simple and quick, but in electron microscope, it is a complex and time-consuming process. In light microscope, images are usually colored. They are naturally colored, or often we also use stain to enhance the image. While in electron microscope, we have a black and white image. So here I add the example. Electron microscopes have high resolution power because electrons have a much shorter wavelength than visible light. As I already explained it, this is an important tip for exam. So you have to remember it. So now let's move towards the another topic, which is a unit conversions. Okay, students, as you know, cells and microorganisms are extremely thin, so we use smaller units to measure them. Means we use millimeter. Millimeter is like that thickness of pen tip. We can say that thickness of pen tip. And micrometer, we use micrometer, and it's just like actually micrometer is the 1,000th of the millimeter used to, and it's actually used to measure cell. Okay, the next one is nanometer. Nanometer is actually the 1,000th of a micrometer, and it's used to measure the virus or molecules. It is extremely smaller. So here is a key conversion. We have this relationship. 1 millimeter is 1,000 micrometer. 1 micrometer is actually 1,000 nanometer, and 1 millimeter is 1 lakh nanometer. So, and you can think it as a chain: millimeter, micrometer, and nanometer. And at each step you have to multiply it with 1,000. Means if you have to convert millimeter into micrometer, you will multiply millimeter with 1,000 and get micrometer. And if you want to convert micrometer into nanometer, you will multiply micrometer with 1000 and you will get nanometer. So here example 2 millimeter is equal to 2000 micrometer and 5 micrometer is actually 5000 nanometer. Okay. So now moving towards the Cambridge exam practice question. So what is question? This type of question are actually mm, often asked in the exam. So we have to we will explain. I will explain this type of question in upcoming videos. Here I give you an example, quick example. Okay, now let's discuss what is question. A micrograph of a mitochondria has a length of 50 millimeter. The printed page. The actual length of the mitochondria is 5 micrometer. Calculate the magnification of the image. Now here we have to convert the magnification of the image. So here what we have to do, first step is converts unit conversion. First we have to convert the micrometer into millimeter because when we have to solve this type of question, both values must have same unit. So here we are going to convert micrometer into a millimeter. And here is a tip to remember: when you go smaller in units, multiply by 1000, and but whenever you go larger in units, you have to divide it by 1000. Here we have to convert smaller unit into a larger unit, so we have to divide it by 1000, and you will get 0.05 nanometer millimeter. Okay. This is the first step. We have to convert the units because both values have same units. Okay, the next step is life formula. Magnification is equal to image size divided by actual size. So here 50 by 50 is an image size millimeter. Okay, and divided by 0.05 millimeter. And when we divide it, we get 10,000. So the final answer is 10,000 x. And remember, you have to place this sign because it is the sign of magnification. Otherwise, you will lose your marks. Okay. So this is all about today's video. I hope you understand it well. If there is any question, leave in the comment. I will discuss. Yeah. Thank you very much.